us the blue marker? Here we go.
Ready, set, go.
So the first thing that happens is the dendritic cell will come from the infected tissue and enter the secondary lymphoid organ through afferent lymphatics, and it will have already phagocytosed a antigen and processed it on MHC molecules. And just pretend those are there, but the red dots are the antigen. And for example, this is a bacterial antigen, and it will have brought with it also other bacteria with it. First thing that happens is that the T cells, and blue is the T cell, they will bail out.
Can't even see it on this video.
T cells are going to be red now too.
Okay.
T cells will bail out of the high endothelial venules, and that's them. And through their T cell receptor and on the MHC, they will make contact with their antigen and become activated. And then once they're activated, so here's the second part. B cells will then bail out of the HEVs and come in contact with their antigen, and their antigen does not have to be expressed on MHC, but they'll make contact with the immunoglobulin, with the whole bacterial cell, like a carbohydrate, for example. And at this point, it'll phagocytose the bacteria and then present, so it'll go back to the T cell here. So here's the B cell. And it will present a, like its cognate protein on MHC to the T cell. And then this is called a conjugate pair. Also, they're connected by a cognate interaction. So what bound here is a different epitope than what the antigen that is being presented on the MHC by the B cell. So the T cell that is already activated will activate the B cell, and they will move over here to the medullary cords. This is the T cell zone, this is the B cell zone, and this is the medullary cords. And the conjugate pair will proliferate and will make a few copies. And this is the primary focus. These are T cells, the red are. B cells are green. And the T cells, by their cytokines, will cause the B cells here to secrete IgM, which will not show up, but I'm going to say this red one again is IgM. And they can only
right IgM.
Right IgM. They can only secrete IgM because they have not undergone somatic hypermutation also, so they're low affinity. And they also haven't undergone isotype switching, which occurs in the germinal center, which we'll explain in a moment. So at this point, we have a primary follicle, primary focus, and then this conjugate pair will then move over into the primary follicle, and this was what the beginning of a germinal center, and then this is kind of a larger view. So over here again, in blue, these are centroblasts. The centroblasts are basically the prodigy to the B cells, which are in green here, that had interaction with the T cells. And then on the outside of here, this is overall is the germinal center. We have the mantle, which is made up of T cells, and then we have follicular dendritic cells, which are derived not from lymphatic cells, and they secrete growth factors, and also they have icosomes, which are basically, they hold on to different types of antigens where the B cells can interact with them. So here we have centroblasts. They've already come in contact with the T cell, and at this point they do not express any surface immunoglobulin, and at this point they are, by the T cell interaction, cause them to now they're undergoing somatic hypermutation by AID, which was activated by the T cell interaction. And also, they are proliferating very rapidly, which this proliferation is driven by growth factors such as BAFF that is secreted by the dendritic cell, follicular dendritic cells. When the centroblasts are, once they proliferate and divide, they give rise to centrocytes, which now again are green. And at this point, the centrocytes move out of the dark zone, which is where the centroblasts are, and they have already undergone somatic hypermutation by the means of the enzyme AID, and they've already undergone isotype and class switching. So at this point, they compete with their immunoglobulin, their surface receptors, for contact with the follicular dendritic cells. And once they do, they will bind to antigens that are being kind of basically contained by the follicular dendritic cells. And then they will, again, phagocytose it by receptor-mediated phagocytosis, and then they will process the antigen as a protein, peptide, to T cells. And the T cells are in the mantle and also throughout this part. But this is basically where the last step goes. They move further out. So it starts in the center, somatic hypermutation. They now express an immunoglobulin receptor, make contact with the dendritic cell, phagocytose an antigen from the dendritic cell, and then process the peptide to T cells. The T cells then, so these are basically the competition step, and mostly really where they compete to bind with their antigen. Because after somatic hypermutation, they can either have a higher affinity or they can have a lower affinity. And at that point, if they have a lower affinity, they die through apoptosis. And if they can outcompete the others and they bind their antigen successfully and make contact with the T cell with the second interaction, they will become activated to become, and the second interaction causes them to become plasma cells, which, unlike these first ones from the primary focus, these plasma cells can secrete different isotypes. So they secrete IgG, IgD, and so on and so forth. And then the plasma cells bail out of the secondary lymphoid tissue through the efferent lymphatics. And that's where they drain back into the blood to home to the infected tissue.